Hi, I'll be talking about protein synthesis in malarial parasite Plasmodium falciparum, which I see as a hurdle race. Malaria is something that is caused by this organism known as Plasmodium falciparum. I'll talk about the special process of protein synthesis in this parasite. So before moving on to this parasite, let's talk about what is the central dogma of life. So the central dogma of life revolves around this molecule known as DNA, which you must have seen in a lot of scientific movies as well. So this DNA is actually encoded form of information inside our cells. This is formed into a molecule which is known as RNA, and then this RNA is used to make protein molecules. So protein molecules are something which are essential part of life. So what you can see is a slide that has this molecule which is known as hemoglobin. So hemoglobin is something that is found in our RBCs, the red blood cells. The other type of protein is this soy protein that we eat. And another type of protein is this insulin molecule, which a lot of diabetic people, they have insulin molecule, inject the insulin molecule in their body to get the, to solve the diabetic issues. So in short, protein is something that is essential for life to exist. And hence the process of protein synthesis is also important. So coming back to my organism of interest, which is Plasmodium falciparum, the process of this protein synthesis in this malarial parasite is actually a kind of a novel, it uses something special, some special mechanisms apart from the normal scenario. So what happens in the normal scenario? What you can see on this slide is a part of this RNA that I talked about in my central dogma slide. So this yellow thing is something which helps the formation, the protein, which helps the formation of protein from this RNA molecule. So this ribosome, it attaches to the RNA and then it scans for the coding sequence, the sequence that can actually make the protein. So once it finds the coding sequence, it makes the protein and then it moves on to form the protein molecule. But somehow, if there is something which is known as upstream ORF, it can actually hinder the process of this protein synthesis. So in case an upstream ORF is present, it can actually, the ribosomes will actually form the protein on this upstream ORF, and then they'll dissociate, thereby not going to the coding sequence and forming the protein. So in this way, they act as a hurdle. So if you are trying to race and there are a lot of hurdles in between, then you'll be deferred to reach your destination. So this is what the ribosomes are facing here. And what we found out in Plasmodium falciparum is that there are 11 upstream ORF per RNA. So this is really like a hurdle race that the ribosomes are trying to reach the coding sequence to form the protein, which is essential for life of this parasite, but then it is being hindered by these upstream ORFs. So what we found is that this parasite is using some special mechanisms to make their proteins in their cell. So one of the special mechanism is leaky scanning and the other is reinitiation. So by using these special mechanisms which are widespread in the parasite is able to make its life going and functioning. However, what we can do is we can utilize this special mechanism to make novel anti-malarial drugs so that we can counter the resistance that is developing in the parasites. So this is what my research is about. Lastly, I would like to state that Indian Science Fest has permission to post my video created for this Talk Your Thesis competition online on its YouTube channel, website, or social media channel publicly. Thank you.